सही है चले so I think it's better कि हम लोग face to face होते हैं but due to situation so I will try to go through the very important topic which is general bacteriology and pathogenesis of infectious diseases. So it's a huge topic, but I will try to summarize it. The most relevant thing, the relevant in a sense that it will has the clinical implication as well. So when you go for pathology questions, so you should know the clinical relevance of all these things, because all these things we have already done in our undergrads teaching sessions, but at that time we are unable to understand the clinical implication and clinical use. So I will go through the slides and I will touch the basic things, but I will try to give some examples related to the clinical aspects as well, and you are open to raise the hand and ask the questions. So there's no formal session, you can interrupt in between. So as I have told that we discussed basics about what is prokaryote and what is the difference between prokaryote and eukaryotes, and we will discuss the anatomy of the bacteria and the important function related to that structure of the cell. We will just touch what is taxonomy, what is classification, and how it helps, and what is the significance. We will discuss the bacterial physiology, what are the important functions related to that specific structure. Pathogenesis of infection, what are the basics of pathogenesis of infection. So you can have an idea about all these things. So it will be all about basics. So I start with on introduction on the base, like when we divide the cellular structure. So you know that there are many types of like trillions of types of cellular structures if we talk about ourselves. So and plants or animals, so it's more organized, it's more developed cellular structure as compared to the native and the old or the primitive sort of cells which do not have very organized and as compared to the more developed cells. So on the basis of that, we can say that the pro means primitive, karyote means cells, as we know that, eu means the new and more developed karyotic cells. So just the anatomy and the structure of prokaryotic cells. So it has a basic structure of cell wall, cell membrane, which restrict and the shape and the size of the cells. Then it has the cell and the liquid portion, which we call it cytoplasm, which do not have so much organized structure and which contribute to the metabolism. So as compared to eukaryotic cells. And locomotion, these cells have a special structure which like flagella, villi, which help them to get attached to the surfaces, which help them to in their locomotion and their movement, which is the important virulent factor for these cells. So these are the structure. We will discuss with details in next slides. And this is the eukaryotic cells. You can see in this. You can see here that these are the structures which are more developed. These are the structure which is more developed in eukaryotic cells, like this endoplasmic reticulum, like the nucleus is more complex, more organized, more structured. So the ribosomes, the endoplasmic reticulum, these are it has more structures. So it has very important organelle, which we call it mitochondria, which is absent in this prokaryotic cells. It has microtubules, which again have a very important role in structuring these cells. It has the Golgi bodies, which is again very important structure in their metabolism. So So they have a common cell membrane, all these things. So this structure, prokaryotic cells and the eukaryotic cells, in comparison, prokaryotes are underdeveloped. So if we go through the structure to structure comparison. So in prokaryotes, the structure is simple, as we know, and we discuss the prokaryotic cell. In comparison, the eukaryotic structure a little bit complex. In prokaryotes, there's no prominent nucleus. We call it nucleoids there. And in eukaryotes, the structure is the prominent nucleus in eukaryotes. The structural size is in prokaryotes is micrometer with comparison to eukaryotes. It has a larger size. Peptidoglycan, it's an important structure in the eukaryotes. So this structure we use it for diagnostic purpose as well. We will see that this peptidoglycan layer is being used to classify them on the basis of peptidoglycan. We divide it the bacteria into gram positive, gram negative. So this is a very important structure. We have to keep it in our mind. But in eukaryotes, the cell wall, it has more structure, which is composed of some cellulose, chitin, which include fats, which include more carbohydrates. So the structure is more tough in eukaryotes. So the ribosomal structure is small as compared to in prokaryotes as compared to eukaryotes. So eukaryotes could be unicellular or multicellular. It forms tissue, it forms more structured systems. So in comparison, prokaryotes are usually unicellular. So organelles are not is usually membrane bound. So it has a structure which is more membrane bound in eukaryotes as compared to organelles, which are loosely dispersed in cytoplasm in prokaryotes. And we know that we have to discuss the prokaryotes here. So main prokaryote are bacteria, which are important component of this universe. In comparison, eukaryotes, which are human, plant, fungi, and protists. So these are the differences in the structure of pro and eukaryote, but there are certain common things, like both contains the genetic material in the form of DNA. So ribosomes are common in this in their structure. They do have cell membrane, which is essential for maintaining the structure, and they contain cytoplasm. So cytoplasm, cell membrane, and the nuclear material are the common factors in both pro and eukaryotic cell, which is essential for to maintaining their life. So just a brief about the how we have started to see this tiny little creatures. So a Dutch scientist name is Leeuwenhoek. He was the first who invented this microscope, and this is the first structure here. This is here is the lens, and you can see that how he is seeing this the tiny microscope. And in I think late 17th century, he was able to identify the different structures on the basis using this microscope. So here you can see that there are different structures of so they were able to identify that initially, but we were thinking that it was some natural disaster that caused this infection and all these things. But they come to know that there are some creatures like these structures they have identified first in late 17th centuries. And now we have different type of microscopes which evolved through different evolutionary process. So in the labs we see this type of microscope, we call it light microscopes, and there are different structures. If they use two lenses, we call it binocular microscopes. If it's a single lens, we call it monocular microscopes. And on the basis of that, we have modified the structures of the of the microscopes. For example, if we tag the object with some fluorescent material, fluorescent objects, so we can see it's under the fluorescent microscope. So it has a fluorescent. It has a device which emit transmit the fluorescence, and under the microscope we can observe it under the like a fluorescent material. So we have different example. We see a fluorescent technique we use in bacteriology, for example. We use fluorescent technique for the identification or for visualizing the spirochetes like Treponema pallidum, like we call it syphilis. So we see Treponema pallidum. Hello. Hello. Can you hear me? Am I audible? Please, can you mute if you don't have any questions? So these type of microscope we use it in. So these type of microscope we use in the lab. These microscopes, but and we use light as a source of source for imaging. But if we have fluorescent, we use it fluorescent. Usually in the lab, we use fluorescence for different objects. We use fluorescent for we're seeing the some fungus also. So there are different microscopes. We use inverted microscopes. We use to see the cell cultures like viral cultures, sometimes bacterial cultures as well. So this type of microscope again, but the source is the light. Light is the source. And now we are moving toward the more automated imaging system. They use the machine learning to analyze the images. And there are other types of microscope, which there are many types of microscopes. So I have just highlighted here this stereo microscope, this digital microscope. Digital microscope, they take the images and they compare it. So in the clinical laboratories, we do not have so much facilities to use the digital, but now we are moving towards this stereo microscopes and digital microscopes. So just to give a flavor about different type of microscope, I have just showed a picture. And this is the basic structure of microscope. So it has eyepiece. From here we evaluate and we see, and it has a tunnel like eye tubes, and these are the main object microscopes, and this is the different objective lenses. We use it for 100 for the and we use 20, 10 magnification, and these are different coarse focus, fine focus. So we use this microscope in the laboratories, and these are the source of light. So light or lamp is placed on the microscope, which reflect the microscope through mirrors, and this is the object we see under the microscope, and this is the objective lens which magnify the image, and from here we can see the we can see the object. Okay, so this is the brief about the microscope. So main is the lens, and the light is the source of light. These lamps are the source of light. So this is the plate. For example, if we see that this bacteria, this is a plate. Sorry. So this is the plate. If we see, so this is a clad plate, and these are all bacterial colonies. We see with
Recording in
if you want to
progress.
see more detailed structure like protein, like small molecules, like atom, like lipid, so we have to go to electron microscopy. So electron microscope is very important, but for the clinical microbiology, we have light microscopes, and these light microscopes we use to visualize the bacteria. So when we see under the microscope, we are light microscope. We cannot see the organelles. We cannot see but we can see the structure of large structure of the bacteria. We can see the shape of the bacteria. So these shapes is very important to classify the bacteria. Like in next slide, we will see. So the shape is the arrangement of the bacteria and the shape of the bacteria is very important to classify in clinical perspective. So when we see the bacteria under the microscope, we can see the shape and the arrangement. So, for example, if someone is calling you in a clinical lab, and when you will go into your practices, like your, for example, if you are doing a surgery and you found an microscope or call from the microbiology resident come to you, and you will see that and they told you that okay, so we are growing gram positive cocci. So you have to realize that what is gram positive cocci, and or someone is telling you it's bacilli, so you have to understand what is bacilli. So these are the shape and arrangement which help in identifying the bacteria, and on the basis of that, we start the antibiotics. If someone is telling you it's cocci and gram-positive cocci, and patient has sepsis and bacteremia, so thing comes to your mind is Staphylococcus aureus. It could be Staphylococcus aureus. So you will go and try on the basis of clinical findings. You can start vancomycin, you can start cloxacillin, like this. So this is very important that when we talk about the cocci, we will tell that which type of cocci. Is it in chains? Is it in clusters? Is it in large clusters? Is it in small diplococci? It's in small chains. So whatever the arrangement, we have to be very clear, so that it's a cocci, so in which arrangement. If it's bacilli, what is the arrangement? If it's the spiral rods, it's a spiral speed or curves. So how many curves is that? So it's very important. So whenever we call about cocci, ask about which type of cocci. Is it gram positive? Is it gram negative? Is it gram positive? So it's in the clusters or in chains. If it's a gram negative, it's in the gram diplococci. So it has some capsule around it. So you have to be very clear कि ये किस चीज की arrangement है. So that was I was talking about. So if it's a diplococci, so which type of diplococci? Is it in lancet shape? So it's a pneumococcus. If it's a diplococci without lancet shape, it could be enterococcus. If it's a coffee bean shape or kidney bean shape, it could be gram negative. It could be Neisseria species. So urethral swab, you have sample of urethral swab, and someone is calling you and telling you it's a gram negative diplococci. So thing comes to my mind is Neisseria. So if it's sputum sample and someone is calling you and telling you it's a gram positive diplococci in lancet shape, so it could be pneumococcus. So if it's in CSF gram positive diplococci, it's pneumococcus. You have to go suddenly it's pneumococcus. So if I told you that if it's in tetrad, it could be staph and any other things. If it's a packet of many gram positive cocci, it could be micrococcus, which you usually consider as contaminant, like this Sarcenia and micrococcus. But if it's streptococci in brain abscess, so it's very important that you delineate that it's streptococci, is streptococci or it's something else. Say if it's this streptococci in urine, so you have to you should be very clear that it could be beta hemolytic strips. So this structure is very important, and whenever someone told you cocci, so ask them whether it's a gram positive or gram negative. The next question will be arrangement. Is it in diplococci? Is it in tetrad? Is it in chains? It's a small chains. It's a gram negative. So what is the arrangement? What's the shape of that gram negative cocci? So that helps you in differentiating the different species. So bacilli could be many, arrangement could be many types. It could be thin, it could be stacked up, it could be palisading, like if it's palisading, it could be coroniforms. If it's stacked up, it's very closely arranged, it's a mycobacterium. If there's a spore, so it's very important to highlight if there's a gram positive bacilli, so whether the spore is present. If a spore is present, what's the arrangement of the spore? It's a terminal, it's a subterminal, it's in the center. So whenever someone told you that it's a gram positive bacilli, so you have to ask which specimen is gram positive bacilli. If it's in the if the arrangement is like this filamentous, just like the filaments. So this is actinomyces, it's nocardia. So whenever you found the bacilli, you have to, again, I will repeat. So whenever you got the cocci and bacilli, you have to first go through किस किस्म के हैं, is it gram positive or is it gram negative. So पहला सवाल ये होगा gram positive or gram negative. Number 1. Number 2, उसकी arrangement कैसी होगी. Is it if it's bacilli, so is it in filamentous, or it's in arrangement is palisading. So this could be your second question. If it's cocci, so what's the arrangement? So I repeat, gram positive, gram negative, and then the arrangement. And the if it's gram negative or gram positive, so is it curves or not? If it curves, so for example, Vibrio, it's curved bacilli. So stool specimen, we usually see like if there's a difference, if we want to differentiate, so there Vibrio has a structure which is curved. Campylobacter, it has seagull wing appearance. So on the basis of gram stain, we can easily differentiate these curved gram. It's spiral shape, so it's spirochetes. These are the different spirochetes. So arrangement, nature has given bacteria a very typical arrangement, and we have to utilize in our clinical practices to upgrade our knowledge and to upgrade the clinical findings. So I repeat, हमें gram stain पहले करना है, and then whatever the gram stain finding, on the basis of that हम उसके arrangement बताएंगे, and it helps you in clinical correlation very well. So I start with the anatomy of the bacterial cell. So broadly हम prokaryotes पे सारा हमारा काम हो रहा होगा अभी अगले next slide. So अब हम prokaryotes की बात करें, so basically हम इसको 2 से 3 चीजों में we divide it into two or more things, like two component, the outer layers which has different components I will discuss, and the fluid portion within the cells, intracellular, and the other additional structure which could be on the surface, on the cell wall, or within the cytoplasm. So the outer layer, it has two components basically, the rigid cell wall and the plasma membrane, which is just beneath the cell wall. And the fluid portion, which call it cytoplasm, it has different inclusions, ribosomes, mesosomes, nuclear material. So if you see this is this outer layer, which includes cell wall. I just ignore capsule for a time being, but if we see this cell wall, these are the plasma membrane or cell membrane, and this is the cytoplasm. So this is the outer layer. It also includes a capsule. Sometime when bacteria is living in a tough environment, so this is the capsule which helps them in the survive. So some organism has capsule, and this capsule we use it for identification feature for as well. For example, this is a capsule which do not permeate the dye to invade, and this helps us in identifying different microorganisms. For example, For example, pneumococcus. Pneumococcus has a very tough capsule, this capsule we can observe in the gram stain findings. Cryptococcus neoformans, which is a fungi, it has a capsule, and that capsule helps us in staining and negative staining, and that can be used in the diagnosis of these microorganisms. So capsule has again a very important features. If we go into the cytoplasm, it has very different structures, and the nuclear material inside is not, do not, if you see that this nuclear material do not have a very compact or closed system. So this is loosely arranged and present in open environment. And the additional structure, so at the additional structure, we also there are plasmids, which is again nuclear material, slime layers, capsule, flagella, this I have discussed, and the fimbriae and pili. These fimbriae, pili, and these pili, these help bacteria in getting attachments. So we'll see later. And these spores are additional structures. So not a very complex structure. Outer layer, which has cell membrane, cell wall, and followed by cell membrane, and it has a cytoplasm, which is again it has different component. So I go into details of these cellular structures. So I start with the cell envelope. So we from the outer layer of cell membrane and the periplasmic space. So we will see what is periplasmic space. So outer membrane, cell wall, periplasmic space, and the capsule. As a whole, we call it cell envelope. So envelope include we go from outside to inside. So some has capsule, then the cell wall, and then the outer membrane, and the periplasmic space. It has a very important role in the disease process, in diagnosis of the bacteria. So if we concentrate on gram-negative and gram-positive bacteria, so if we see one by one, so if we see gram-negative bacteria, which is very important component, and it is very important in causing different diseases. So if you see this structure, so it has an outer membrane which is missing in gram-positive organism. So this is the outer membrane, which contain different types of porins, lipopolysaccharides. These channels helps in the transport of essential materials, transport of antibiotics, and mutation in these porins, mutation in these structures helps them in survival, in their survival. So these are the outer membrane, which is essential component of gram-negative. These are the periplasmic space, which contain different enzymes, different virulence factors that help them also to survive in different against different antibiotics. So these are the periplasmic space, which contain different enzymes, this periplasmic space also contains this peptidoglycan layer, and this is a cell membrane which is common in both these things. If you see that this periplasmic space, it contains a peptidoglycan layer, which is very important in if you see this peptidoglycan layer, in gram-negative rod it is very thin, but in gram-positive bacilli, if you see gram-positive bacilli, it has a thick peptidoglycan layer. So this peptidoglycan layer is very important in identifying the bacteria. So if you see gram-negative, this is very thin, and in gram-positive it is very thick. And when we do gram stains, so this peptidoglycan layer helps in differentiating the gram-positive and gram-negative. So this thing we have to learn, and we have to remember these things. Apart from this peptidoglycan layer, if you see gram-positive organism, these are the teichoic acid and lipoticoic acids. This is not very common and not prominent in gram-negative bacteria, but in gram-positive bacteria, this teichoic acid and lipoticoic acids is very important in different virulence factor, and we see that mycobacterium and different other types, which is weak gram-positive organism, this teichoic acid helps them in staining as well. So this is very important in gram-positive and gram-negative. So I will not go into more detail about the mycobacterium. Just have a look that this is fungi and this is so this fungi and this mycobacterium structure. So this is relatively it's different from this gram-positive bacteria and gram-negative bacteria. Here the mycolic acid is very prominent. Here this glucan and this menan is very prominent. And this menan and glucan, we use it. I think Dr. Javeri has covered this thing. That this will be used in diagnosis of this fungi. Here, this mycolic acid is used for the identification of this mycobacterium. So cell wall structure, I repeat, the cell wall structure is very important in classifying the bacteria on the basis of peptidoglycan thickness. We classify gram-negative and gram-positive. Gram-positive has thick peptidoglycan and gram-negative thin peptidoglycan. So cell wall, if we start from the cell wall, which is an important structure, which is the outermost layer. If some bacteria do not have capsule, so this is the outermost layer. And we see that the outermost layer enclose the cytoplasm. So what's the purpose of cell wall? Cell wall agar na ho to kya hoga? So the shape and the structure and rigidity we do not of bacteria we cannot do not have a bacteria. So it gives the shape and rigidity. It has different thickness and it's a component composed of polymers, polysaccharides, polypeptidoglycan, which is composed of this N-acetyl glycosamine and N-acetyluronic acid. It's alternating chain, and this is alternating chain held by different peptide chains. So these are the pentapeptide chains, and it's connected with different and it forms a lattice network like this. So we have to remember, we have to just remember this thing, that this peptidoglycan layer has a clinical significance. It different bacterial antigens. It contains the different virulence factors. It contains different immunogenic factors. So and the mutation in this structure leads to antimicrobial resistance. So this structure is very important. जो हम बात कर रहे थे peptidoglycan, on the basis of that we divide it into a different structure. On the basis of this structure helps in use as a source or mode of antibiotics to act. So it has a receptors. So it has many virulence factors. So the cell wall structure is important, and it helps in different giving shape, not only shape, but helps in giving the virulence to this pathogens. It's a site for different antibiotics, and mutation in this peptides and structure helps bacteria to survive by antimicrobial resistance. So हम जो antimicrobial resistance की बात कर रहे होते हैं, तो जो one of the factors जहाँ mutations हो रही हैं वो इन structures में mutations हो रही हैं. So हम जब भी बात करेंगे कि bacterial structure के mutations हुई हैं, so this is one of the important site for having resistance, for having developing resistance. We have seen in our last slide that these are the outer membrane. These are the outer membrane, which is outside the gram-negative bacteria, which is a very important site. Which is very less in gram-positives, almost negligible, but it's very common, very prominent in gram-negative organisms. And it is more, as I told you, there's no periplasmic space and no significant outer membrane in gram-positives. But if you see the gram-negatives, so this is, for example, it's a gram-negative intact outer membrane. It's a major permeability barrier for the nutrients, for the antibiotics, for the other factors. If you see, it's very intact, it's very important, this divalent cation, which cationic and this hydrophobic and hydrophilic structures. So this structure is intact, and in between this in its outer and inner membrane, there's a periplasmic space and stores different enzymes. सही सो अगर ये इसमें कहीं इसके स्ट्रक्चर में कहीं डेविएशंस आती हैं, सो वी हैव सीन दैट ग्राम नेगेटिव organism have more resistance, they are more their nutrition requirement, their metabolism is more complex than the gram positive organism. So if someone is treating with colistin for a long time, so this colistin, you can see the structure, the bacteria, and this structure, colistin treated structures. They develop modification in their cell wall, and this helped to this bacteria to have more fluid to go outside, to have more external factors to come inside and help them to in more mutation, in developing more mutation. So antibiotics affect the outer membrane. Just we have an example of colistin. And if you can see this in this structure, due to many factors, this symmetry which we see here is lost. So there are many mislocalized polysaccharides in between, and due to this, we have seen that this is the only anterograde transport, but now it's both side transport, anterograde and retrograde. And due to this, bacteria lost their structure and they develop different, allow different virulent factor to come and go inside. And this is again a very, you see this structure modification in these outer membrane. So outer membrane is a very important structure clinically. Due to different antibiotic and different drug exposure, it lost their structure and has developed the resistance. So I've seen that you have seen that in gram-positive organism, the cell wall is very important. So if you see the gram-positive here, so gram-positive, I repeat, gram-positive, a thick cell wall, and this is the thick cell wall. So gram-positive organism retain the crystal violet. When we do the gram staining, it retains the crystal violet. In comparison, gram-negative has a very thin peptidoglycan layer, and therefore, when we decolorize it, it easily decolorize and followed by the counter stain, which take the counter stain by safranin. So cell wall has a very important structure, which is peptidoglycan, and it divides the organism into gram-positive and gram-negative organism. So there's a transport across the cell membrane, which is called downhill movement. If this cell membrane allows bacteria to have nutritional antibiotics, and all these things move across this cell membrane. If it's from a higher concentration to low concentration, so it could be through diffusion, and it could be through passive transport or facilitated diffusion. But if it's uphill means from low concentration to high concentration. So for that, it's uphill. So we know that for uphill, we need to have more ATPs, we need to have active transport, and for active transport, it needs energy. So there are different pumps which helps uphill movement in their cell membrane and cell wall of it. So this cell wall is not only given the structure, but it also have this active transport channels, which allows different nutrition, which allows antibiotics to come inside. So there are different drugs which use this transport channels, and these drugs act on this active transport and inhibit this and block this transport channels, which allows these antibiotics to work. So when we come from outside to inside, we have discussed our cell membrane, we have discussed outer membrane, we have discussed the clinical uses of that, and then followed by the cytoplasmic membrane. This is again it's compared to cell membrane, it's a very thin layer and separates cytoplasm from the outer membrane. And it's a semi-permeable membrane. Previously, it was it develops the permeability due to different cell wall structures, and it controls the inflow and outflow of the metabolites. So if it's permeable to everything, so the metabolites inside the cell, inside the bacterial cell, it will go very easily outside. So to retain the metabolites and the nutrient structure, this is a very important plasma membrane. And it's composed of lipoproteins and a small amount of carbohydrates. So you see this polar structure. It has phospholipids and a cholesterol in between. So these are the different carbohydrate chains on the surface of that. So cytoplasmic membrane is a very important component. Um, so if we go from outer, then cell wall, capsule, cell wall, plasma membrane, then comes the cytoplasmic component. So here, cytoplasm is not very complex. It has different. It has few things like ribosome, which helps in protein synthesis, and many antibiotics like tetracycline, like like macrolides, they act on this and inhibit this protein synthesis. So there's a mesosome, which is an invagination of this plasma membrane, and it's a site for respiratory enzyme to work here, and it coordinate with nuclear and cytoplasmic division during the biodiffusion. So mesosomes have their own usage, and there are many intracytoplasmic inclusion bodies that helps bacteria to reserve the energy in the form of phosphate bonds, in the form of different granules, and these cytoplasmic granules is also used for identification of bacteria. For example, metachromatic granules in the diphtheria bacilli we use as a features to identify the bacteria. When we stain it, we stain these metachromatic granules, and the gram-positive bacilli, which is a common source of diphtheria, Cranibacterium diphtheriae. So when we come across the sample in the laboratory, we see that this metachromatic granule is used for identification. So as we know that the nucleus is not very structured, and we have nucleolus, no nucleolus and no nuclear membrane. So we call it nucleoids, just like looking like nucleus. And the chromosomes are present in loose forms. And there's another factor, we call it plasmid, which contains the extranuclear genetic material. And this plasmid is not necessary for the life, but this plasmid is very, very important because it carry essential information from one cell to another cell through different genetic transfer or by diffusion. So this plasmid carries the plasmid gene for antimicrobial resistance, and it transfers from one bacterium to another. It carries the drug resistance and toxicity genes also. So remember this plasmid. We nowadays the scientists are working on plasmids, and these plasmids are used for transfer of antimicrobial resistance. So you have to remember this plasmid is a very important essential component. So I'm go very quickly through this. We have discussed that additional organelles which are present on the surfaces are slime layers, capsules. These help in giving the structure to the bacteria, and it has antigenic nature as well. So we use these structures for antigenic determinants, but the important thing that this structure allow bacteria to survive by having more antigenic structure, by inhibit phagocytosis, and we see it. Important thing is that we use it for identification features, like for India ink. Quilling reactions is not used nowadays, but we use India ink and other structure, Gramston sometimes, to identify the structure, and it helps in bacterial identification. Flagella, these are the structures which have different arrangement of different bacteria. So these are basically protein structures, and it helps in identification of the bacteria. And it is also the antigenic determinants like H antigens on the gram-negative bacteria. This helps in identifying the bacteria. It has more epidemiological role. Like for example, Salmonella typhi or Salmonella species, it has H antigen, very prominent H antigen, and this H antigen helps in identifying the serological identification of bacteria like Salmonella, and we use it in epidemiological studies. So this flagella helps bacteria in locomotion, and it's used in bacterial identification in the routine clinical labs as in epidemiological sites. So this again, this structure we call it fimbriae and pillae. So fimbriae are the thin hair-like appendages on the surfaces of many gram-negatives like this, and it helps in adhesions. So these pillae and fimbriae help in adhesions, and with the use of this pillae and fimbriae, they help in colonizing the environment. internal environment in the human and animals, and the external environment on the surfaces of external surfaces uh, the anything, any object on the surface, external objects. So it again composed of different filamentous protein like pili and proteins. So and it helps in transfer of the genetic material. So pili again, so this pili helps in transfer of genetic material, which again include any antimicrobial resistance genes and any other important gene which when they develop trans, uh, during their survival process. Spores are usually these are the highly resistant resting stage and formed during the adverse environment like depletion of nutrients. Formed inside the parent cells and it's called the endospores. So endospores are very important as it survive in the tough conditions like radiations, high and low temperatures. Uh, as it difficult to kill during sterilization, so these can be helpful, this help in survival as it do not get sterilized and kill in low temperatures. So it can easily be cause infections like we know that the bacillus anthrax, bacillus cereus, it causes food poisoning, and clostridium tetani, which is called tetani, perfringens called gangrene, and botulinum. So we see these patients if we do not sterilize the things, or we do not uh, use the unsterile instrument, so there's a chance that we got the tetani, clostridium. We transfer this infection to our patients. Uh, if we do not boil and we do not heat the foods, we get the chance of food poisoning. So so these are the important organelles we have discussed, the cell wall structures. Um, so now I move to the another important component of the which we call it taxonomy. So taxonomy is why taxonomy is important. So when we call it taxonomy, it has different components. So I will touch the very important things, which is important to know is the classification. So we classify the bacteria is the to have an orderly grouping of microorganisms. So we call it gram positives, we call it gram negative, we call it lactose fermenter, we call it non-lactose fermenter, we call it cell wall, we call it cell wall structures, without cell wall structures. So we call it we can name it different on the basis of so we classify orderly grouping of these organisms. So there are different things. Broadly, we call it phenotypic methods, like on the basis of cell wall composition, like we call it gram positives, gram negative, we call it mycobacterium. On the basis of cell wall, we have discussed right now. And the basis of biochemical features, we call it nutritionally variant, we call it lactose fermenter who requires more lactose for on the basis of biochemical features, their nutritional requirements, divide again. So there are different methods. We classify organism on different methods. But now it's more evolutionary process. We now classify the bacteria. We change the classification of the bacteria. We reorganize the bacteria on the basis of the evolutionary process, like their DNA composition. Initially, it was the DNA compositions, then 16S ribosomes. We check it on the different methods by checking the 16 ribosomes, and the whole genome sequencing. Now we are, and you will be facing the whole genome sequencing. So the next century we will talk about the whole genome sequencing. So phenotypic methods probably will be redundant in few years. Now we will classify the bacteria, we classify the organism on the basis of whole. WGS means whole genome sequencing. Whole genome. So we sequence, we arrange, we see the sequencing of the genomes as a whole, and we classify कि ये bacteria, ये viruses, ये चीजें ये हैं तो ये इस group में आ जाएंगी. So now we will go for whole. So you have to remember whole genome sequencing. So nomenclature is the agreement upon naming of the organism. Like we call it staph aureus. So staph is the Staphylococcus is the genus. So and this is aureus. If we call it, someone call me this. There's a growth of staph. So I will be confused that this type of staph. If it's staph epidermidis, so it has a different impact, clinical impact. If it's Staph aureus, it has different clinical impact. So whenever you talk about the nomenclature, we generally call it genus, and up to species level is very important to classify. So now labs also reporting not Staph species. We try to report Staph aureus up to species level, and in some species we call it Salmonella enterica. So like this is genus, this is species, and this is the subspecies. So different species and different. So it has different implications. So basically, we have to give name uh, up to species level. So this is the genus, this is the species, and sometimes we need to have more species level like Salmonella typhi. We are writing Salmonella typhi as Salmonella enterica subspecies enterica. So this subspecies enterica has different or more species, typhi, typhimurium, and all these things. So now this nomenclature has more epidemiological concerns. So if someone telling Salmonella, so we have to ask which type of Salmonella is enterica. So enterica with which type of typhi, typhimurium, paratyphi, all these things. So every subspecies has different epidemiological impact. It has different virulence. It has different. So nomenclature is very important to not only to give name, but it has also epidemiological implication. So if we see clinically, so we rank different bacteria, classify on the basis of their ribosomal DNA sequencing, and we divide like domain, phylum, class, order, family. So important clinical importance, when it's called about clinical importance, so it starts from here. So we have to classify that it's Staphylococcus, it's which genus it belongs and which species. So you see that if we see the similarity on the basis of 16S ribosomal DNA, so it more close in family Staphylococcus more has more clinical, has more similarity like 86%, and if we go to genus, it has more 94%, and if we go to Staph aureus, then it's more close there. All the Staph aureus have more close structure there. So this is the basics of taxonomy. So I repeat that you have to classify bacteria, and then you have to get nomenclature the bacteria on the basis of genus and species. So when we talk about the physiology of the cell wall, we have discussed up till now the anatomy of the cell, anatomy of the bacterial cell. We have discussed many component of the cell structure: cell wall, cell membrane, plasma membrane, capsule, cytoplasm. Then we have discussed the taxonomy and the taxonomy of the classification and the nomenclature. So now we will briefly touch upon the bacterial physiology. So bacterial physiology, when we talk about bacteria, so just like human body, bacterial physiology also require nutrition. It has external environment on basis of which they survive. We give them artificial environment in the laboratory so they can grow on that specific environment. How it grow and multiply, and it has a complex metabolism. So I will not discuss about the metabolism here, but just discuss a few growth requirement. So for the optimal growth requirement, for the growth of bacteria, they need water. Essentially, they need different nutrition, which include carbon, include nitrogen, inorganic salt, growth factors, and source of energy. So growth factors, for example, if we talk about growth factors, for example, Haemophilus influenza, they require growth factors, X factor, NAD factors. Some bacteria, they require more carbon, like Citrobacter, they need more carbon sources for their energy. Some require more nitrogen. So these nutrients are present in the environment, and they utilize different sources of nutrition from different resources and use as for their survival. But in clinical laboratory, we give them all these things artificially. So artificial environment, we give them in the form of agar, in the form of broth, and we give them oxygen, and we give them carbon dioxide to grow the bacteria in the laboratory. So this we have discussed that they take this from the surrounding environment, but in the laboratory we gave them artificially by the form of simple media with different source of nutrition. So broadly, we divide some organism on the basis of the oxygen requirement. Some organism only require oxygen uh, for their survival, like for example mycobacterium. But some organism obligate, like they only grow in the absence of oxygen. The obligate aerobes means they require only oxygen. They do not grow in the absence of oxygen. Some are obligate anaerobes that they do not require oxygen and they only require anaerobic condition. And if we give them oxygen, it will die. So example is Clostridium. But majority of the organisms are facultative anaerobes. Majority which we can easily grow in the laboratory, and they grow generally aerobic condition, but they also grow in the absence of little bit oxygen. So most of the bacteria which we grow in the laboratory are bacteria of medical importance. See? So majority of the laboratory bacteria are facultative anaerobes. So microaerophilic bacteria are those which require low oxygen tension. Which a common example is a Campylobacter. This is the most common example of microphilics. So majority of the bacteria which we grow in the laboratory are facultative anaerobes, but we require, for organism to grow, we should give them a microaerophilic condition, like for Campylobacter, and for the those bacteria which grow anaerobically, we give them anaerobic condition as well. So temperature obviously requires is essential for the growth of bacteria. Majority of the pathogen, as I've told you, is grow in this environment, in this temperature. But the clinically important bacteria which we require, if we give them more bacteria, more temperature, so these bacteria will die. So these are the important bacteria. But this thermophilic bacteria we use, we cannot grow this bacteria, but we use this temperature as an important tool for checking the sterilization process. So again, pH is very important, as we know that for any growth, for any life, pH is very important. So for that, we have to maintain the good growth environment by using different pH. So, but in clinical lab, we use this important pH for the growth of bacteria. Some bacteria are very sensitive to change in the pH, like Vibrio cholera. If we change the pH of this environment, these bacteria will not grow. So maintaining the pH for the growth of the bacteria is very important. Okay, so bacteria has different metabolism pathway, like carbohydrate, protein, fats. They use different channels for their bacterial survival. They use mainly the aerobic bacteria obtain their energy by oxidation involving the O2 and oxygen receptors. So, but there are different resources. They use different metabolism pathway for their survival. So when we talk about these are and when we go into the laboratory, so when we take the sample, for example, so there are different growth phases in the life cycle of the bacteria. So we call it the lag phase, exponential phase, and the stationary phase, and the death phase. So in that phase, when we talk about that phase, so I will start from here. So in the lag phase, so for example, if you take the sample of blood cultures in from a patient, so in patient, if some specimen or we take any specimen, for example, a diabetic foot, we take a aspirate. So probably in that specimen, we have bacteria which could be in lag phase, could be in their log phase. So in the laboratory, when we get the sample, so our target is to bring the bacteria from lag phase to the exponential phase. So we give them different media, we put them in the broth. So in that, we we bring the bacteria and from lag to log phase. So in lag phase, the live cell are very few. But when we give them enrichment, we give them some media to grow, it goes from the lag phase to the log phase. So it's very important that when we take the specimen of the patient, we should maintain the and we submit the specimen as soon as possible to the laboratory, so that the bacteria which are in the lag phase must go into the log phase. And when bacteria goes into log phase, from here we can easily isolate and cultivate the bacteria. So some bacteria take more time from lag phase to log phase. For example, TB, it takes more than uh, 48 hours for going from lag phase to log phase. That's why TB culture takes more time in the laboratory to grow. It takes 4 to 8 weeks. But some bacteria have very quick lag time to log time phases, like E. coli, like Staph aureus. They grow only in the time duration of 3 to 4 hours or 6 hours. So it's important that we maintain this lag to log phase, exponential phase, and after that bacteria in the specimen will go into stationary phase, and it will go ultimately to death phase. So in death phase, the total number of bacteria might be more. So it could be more here. But the viable, means the live bacteria, is the implication of this growth phase. We have to remember that when we go to the specimen and we send the specimen in the clinical lab, so bacteria might be in the lag phase, and it may be in this phase, death phase. So the sample quality and the viability of the sample and the purpose of the laboratory is to convert this lag phase into log phase, and then we can able to isolate the bacteria. So this phase is very important to remember. So, well, as I told you that some bacteria like coliforms, 20 minutes. Say tubercular bacilli, it's more than 20 hours, 48 hours, and the lepra bacilli, it takes more than 20 days. So it depends upon. So that's why it's difficult to grow tubercular bacilli, lepra bacilli in the laboratory. It require more time, it require more enrichment and nutrition. So this is the normal physiology of the bacteria, which you have to consider and remember your exam point of view. So I will go to the pathogenesis. I will take, I'm just about my time, but I think the topic is so lengthy, so I have to cover many topics. So pathogenesis of the bacterial infection. So pathogenesis of bacterial infection. So there are certain terms which you have to remember, but important thing is that the virulence factor, virulence. Virulence is the degree of pathogenicity of an organism, how virulent an organism. For example, Staph aureus has more virulent factors, so it has more pathogenicity in comparison to Staph epidermidis. So virulence for pathogenicity, we have to remember that virulence factor or virulence is very important in determining the pathogenesis of pathogenicity. So not all pathogen entering the human body will cause disease, because humans are protected by normal flora and the immune system. So virulence is a very important thing, just to start with the pathogenesis. So when we talk about the infectious diseases, there are 4 phases or periods in the infectious disease. So whenever we acquire an infection, so it has an incubation period. During this time, the pathogen do not cause infection, but it was just like establishing in the human body. So they are producing more virulence factor. So it's a time between the entry of pathogen and the onset of symptoms. For example, मैंने कोई चीज खाई and I got in diarrhea. So probably the cholera, Campylobacter, which enter into a body, takes time to produce their virulence factor. This time we call it the incubation. So probably 1 day before or 12 hours before I got this pathogen into my body, and it incubated in my body and produced the virulence factor, and after 12 hours I got the symptoms. This is incubation times. Second is the prodromal period, the time when the person feels abnormal, and after that, so prodromal finds वो चीज़ बेहतर महसूस नहीं हो रही है, अच्छा नहीं लग रहा है, चीज़ें तबीयत बिगड़ना शुरू हो रही है, that is the prodromal time. And the period of illness, I started to have my diarrhea, vomiting. So this is the period of illness. Before that, I have small abdominal pain. I have a discomfort. That was the prodromal period, and after that, started to feel some symptoms. This is my typical. And the convalescent, and when I started to recover, in that time, I started to develop the antibodies, and my area of infection started to subside. So these are the four phases in any infectious disease: incubation, prodromal, illness, and convalescent period. Uh, so some infections are localized, like abscesses, small abscesses. But if we do not cure that, or we do not curtail that infection, it could be involved in multi-organ. So we call it systemic infection or localized infection. The disease could be acute, it could be chronic, it could be subacute. So these are different stages of the disease. And the sign, we know that the sign is evidence of disease found, seen by the doctors, and symptoms are the evidence of disease explained by the patient. So steps क्या होता है? सबसे पहले पैथोजन एंटर होगा बॉडी के अंदर, इट गॉट अटैच by through many factors, by capsule, slime, fimbriae, villi, anything. So this attachment requires the receptors as well on the surface of the whatever the host animal, plant, or human. So it colonized there. After colonization, they started to release their factors, virulence factors, and then the pathogen get invade in the human body, and the immune. After that, there will be immune response which inhibit or which progress the disease, and the pathogen also produce toxins. So these are the primary steps in the pathogenesis of infectious diseases. So we can the pathogen can intrude through many routes. So it could intrude through the skin penetration. It could intrude through the mucous membrane, like ingestion, inhalation, through genetic urinary tracts, through different vectors like mosquito bite, like this. Again, it got attached to different, as I told you, that different factors help them in attachment. And it got colonized by different factors, by different environment. Like for example, we have a very good example, H. pylori. H. pylori produce so just about right. So H. pylori is about to survive in acidic environment, produce the enzyme urease enzyme, and it invade. It invade the normal host membrane and the immune systems, and then then our immune response and immune systems would affect these things, and then at the end, bacteria also produce the toxin, different type of toxins, endotoxin and exotoxins. So these are different. So just to sum here, there are different portal of entry to mesenchymal skin, root, mucous membrane. It penetrate, invade our host defense system, and then it damaged by different toxin and hypersensitivity reaction. So at the end, I just want to summarize the things that the thing to remember is the prokaryote has simple structure. They do not have nucleus. Cell wall has peptidoglycan membrane. Bacteria has different shape and arrangement, like cocci, bacilli, and spiral shape. The gram-negative, gram-positive bacteria has thick peptidoglycan layers, and gram-negative have thin peptidoglycan. The transport across the cell membrane to active transport through ATP pump. These bacteria do not have nucleus, but they have nuclei and the genetic material like plasmids. So when we talk about taxonomy, there are different features, but the more important thing you have to remember is the classification and nomenclature. Classification is the grouping, and the nomenclature is the orderly giving name of these pathogen. So bacterial physiology depends upon multiple things, but important thing you have to consider is the nutrition, which includes different factors, environment, which include oxygen, CO2, pH, and all these things. And for pathogenesis, it requires entry, attachment, colonization, invasion, immune response, and toxins. Thank you. So I stop here, and the forum is open for discussion and question and answer.